Hello, my name is Philip Seitzer, and this is the second in a series of tutorials demonstrating how to use the comparative genomics tool JContextExplorer. In this tutorial, we'll review how to computationally predict operons, compare genomic contexts based on factors such as gene order. We'll load a phylogenetic tree as well as computationally predicted promoters, and we'll explore a couple of other features in the context of the cobalamin biosynthesis operon in various halophilic archaea. So in this tutorial, you're going to need a few files which are available on the Facciati Lab website. This is all for a page. And in particular, you'll need the haloarchaea genome set, variable here, the RPOB-based phylogenetic tree, available here, and then this directory containing motif-based computationally predicted promoters for these 80 halophilic archaea. I've already downloaded these files, so I won't do this step here. But once you have downloaded them, you can start JContextExplorer. And now, if we go to genomes, we need to load our genome set. And in this case, we will import a genome set from the .gs file, and that was that haloarchaea link on the website. So here it is, haloarchaea.gs. Click Open. Okay, and now if we go up to genomes, we'll see that we have a genome set called haloarchaea. By the way, we could also have streamed in this particular genome set from the Retrieve Popular Genome Set haloarchaea option. And if we take a look at the contents, we see we have 80 genomes, and we can learn things about each of these genomes. Next, let's load in the phylogenetic tree, which we can do by coming to the Phylogeny tab here in the mainframe, selecting the Load button. This can also be achieved by load phylogenetic tree or Command P. This is the name of the tree, and we see that now the tree appears under the current tree label. I'll change some of these options so that when the tree is rendered it will be a little bit neater. Now let's load those promoter motifs. We can do that by coming to the Motifs tab, clicking Manage Motifs, or Load Sequence Motifs will also work. We're going to load a promoter. And in this case, I will not associate the promoter with particular elements, with particular genes, though you often would. And in these particular files that I've generated were made using the program FIMO, which stands for Find Individual Motif Occurrences, and is a part of the meme suite. And the folder should be called BRE Tata, where BRE is B recognition element, Tata is Tata box. Okay, now that that's loaded, you can click Add and OK. And you'll see that now you have an available sequence motif called promoter. You have a phylogenetic tree called 500B RPOB double prime 80 halos. And you have 80 halophilic archaea. So the next thing I'd like to show you is how to define a context set that may be used often. And this is a primitive way to computationally predict operons. So if you click Add Remove, this first option, Group Genes Based on Intergenic Distance, will serve as our operon prediction algorithm. So what this will do is all genes that have an intergenic distance of less than or equal to 50 nucleotides, so the distance between the stop of one and the start of the next, will be grouped together. will require that they must be on the same strand. And we'll click Add, and we'll click OK. So a detailed description of all of the available context sets and all the things you can do with phylogenetic trees and all the things you can do with motifs are of course available in the user's manual, which you can access right here, also via the Java Lab website. So now we have our context set defined. So I've determined a particular gene, and here I'll show it in single gene, that I'm interested in all genes that are in homology cluster number 1051. If we take a look at what these are, they're all annotated as adenosylcobinamide phosphate synthase, which is a gene well known to be associated with the cobalamin biosynthesis pathway. Okay, so now I'll switch back to my Operons set. So now when I do a search, I will get not only the gene, but also all genes that are within a nearby range. And in this case, there are a large number of genes that are collinear, that are nearby and are on the same strand. So those are also shown. Those come with the adenosylcobinamide phosphate synthase gene. So now we can build a context tree. So I will check Render Context Tree in the analysis options. And since I have a phylogenetic tree loaded, I will also display the results with the phylogeny. Notice I can deselect and select all. So let's go ahead and search. Okay, so search results just as before, but now we have a context tree. Now, this context tree was generated using variable group agglomerative hierarchical clustering. And variable group agglomerative hierarchical clustering is a generalization of the more commonly known hierarchical clustering. And the generalization essentially takes care of any inconsistencies that may arise when the order of comparisons matters. And cases where the order would make a difference are shown by these strange-looking rectangles called bands. So depending on your needs, you may or may not want to show the bands. So let's go ahead and hide the bands for now. Then I'll click Update. And as we can see, now the bands have disappeared. So the tree might look more like something you're more used to. And now we can look at the phylogenetic tree, and we see that the phylogeny is shown with the context tree. Okay, so something that I did not mention in the previous tutorial is you can actually select a number of things simultaneously. So here we see that a number of these organisms are in the genus Haloarcula. So let's select all the Haloarcula. Those are selected in the context tree and in the phylogenetic tree as well. But let's also select the Halotherax. So now you'll see those are also selected. And they are selected here. And once again here. So notice that in the phylogeny, all the Halotherax group together and all the Haloarcula group together. But based on our context tree, it looks like the Halotherax are all grouping together here. But down here, there is some other things happening. So this sort of shows one of the things that you might want to do with JContextExplorer, which is to compare how a phylogenetic tree may differ from a context tree. And when I say phylogenetic tree, I really mean any tree. You can load any tree you want, and it's up to you how you generate it. And just looking at this group, for example, you can do the same thing the other way, where In this context tree, these are very closely related. And now in the phylogenetic tree, you see that they're all over the place. So maybe you want to see what are those contexts. Well, they look like this, yet phylogenetically they look very different. So if you look under this tree band, you'll see there's a huge amount of things you can do. You can change the axis, you can show labels, you can change the node size. I'll just do that to demonstrate. So now the nodes are larger. You can change the labels. And these last few areas, nodes, axis, these are fairly cosmetic. However, this top part, tree computation, brings to light a very important feature of JContextExplorer. So the clustering algorithm is a fairly standard thing amongst all hierarchical clustering type programs. And as you can see, as a default, you're using unweighted average. You can change that. And changing it will, of course, change how the tree looks. But perhaps most relevant to our work here is the dissimilarity metric. So that's essentially the quantity that describes how do you compare these genomic segments. What do you value as important, and how do you, essentially how do you mathematically translate what you think are biologically meaningful differences? So as you can see, there are a couple of default settings, and these are all explained in more detail in the user's manual. But I mentioned that we'd be interested in gene order for this particular operon. So let's make a dissimilarity metric that focuses on gene order. And we can do that by clicking Add Remove here, or also in the load menu, dissimilarity measure, which is Command 2. Let's click Add Remove. And as you can see, there's a huge amount of options. There you have 2 different amalgamation types that you can choose from. And then among that, you can decide which factors are important to you. So let's just, because we're interested in gene order, we're going to focus exclusively on gene order. So we'll check the changes in gene order. And now we have 2 subtypes that we can care about. And let's care about both with equal weight. Click Add Dissimilarity Measure and Submit. And again, a more detailed description of the different factors you can include and the way in which you can combine them to create a customized dissimilarity metric is available in the user's manual. So now we have our gene order, unweighted average. Let's go ahead and redo this search using our new dissimilarity metric. And now what we get, if we look at the tree, is a very clean tree. Before, we had a very complicated tree with lots of different things present and absent. But now it looks like things are much simpler. So to highlight what this tree means and how this matters, I've determined that I'm going to show just 2 nodes stemming from 2 organisms. And now if I click View Context here, so you can see right away that certain genes have shifted. And to exemplify that, here I'll right-click anywhere on the frame, and I'll click Show Legend Complete. And now I can see the annotation and cluster ID mapping and how that relates to this visualized, these 2 genomic segments. So again, I started with 1051, which is this gene, adenosylcobinamide phosphate synthase. And if I just click through, you can see how the genes have rearranged. So far things are consistent, And then you come to this tan gene, and all of a sudden it seems to have moved in the case, from the middle of the putative operon in Halogeometricum boronquense all the way to the head in Halococcus saccharolyticus. And here you can, if we use Control Click, excuse me, we can really see how these segments have changed. So you see these 4 genes are all collinear here, but now there's a big change. And if I really, and I can do a similar thing here and see how in this case these 3 are together and now there's this huge separation. So clearly the genes have changed in their ordering, not so much in their content, just in their ordering. One of the features of the multi-genome browser is the ability to visualize the loaded motifs. So if we go back to our motif frame, we have this loaded motif promoter. So if I check this box, include motifs in context display, now, and then I update context, I can see these promoter motifs appear. I can click on them to learn information, including the specific sequence associated with this motif. And because I did not associate these motifs with any genes, you see they're grayed out. They are considered surrounding, but if I color the surrounding, they all have a color. And again, another feature of the motifs, though I did not show it here in this tutorial, when you make these customized dissimilarity metrics, one of the options is you can talk about how many, or if a gene contains a promoter motif in this case, or it does not contain a motif. So presence absence of common motifs can be used as a factor in determining the dissimilarity between 2 genomic segments. Okay, that completes tutorial number 2. Again, I highly recommend checking out the user's manual for more in-depth about many of the concepts I've discussed here. And thank you for watching.